Bismillah Rahman Rahim. Assalamu alaikum students. I'm Sabir Hasnain, your science subject teacher from grade 8. Welcome back after summer vacations. If you remember, before summer vacations, we discussed about salts, their names, their types, how we give them the names, and there are 4 different methods through which we can get or make salts. In the first one, if you remember, and if you remember, those 4 different methods are by using acid and alkalis, acid and metals, acid and metal carbonates, acid and metal oxide. Through these methods we can make different type of salts. Okay, now in this lecture our aim is to discuss about titration using acid and alkalis. Through acid and alkalis, I mean by the first method we can discuss about the titration. So in this video lecture, we will discuss in detail about the process or the experiment titration. So the question is, what is titration? We know that whenever acid and an alkali react together, we get salt and water. I mean salt and water are produced whenever acid and alkali react together. So the experiment in which we can find out the exact amount of alkali is required to neutralize an acid is called titration. What does it mean? Alkali, I said alkali and acid are added together and gives us salt and water. So definitely we add them by some fixed amount. Let's take an example, although example is a different example. When your mother or your sister make tea at home, then what they do, they actually mix a constant or the fixed amount, 2 cups of water, 2 cups of milk, or 3 cups of water, 3 cups of milk, and relevant to that they add sugar and they add tea in that in water or milk. So now in this case our aim is to neutralize an acid. So to neutralize an acid we must need an exact amount of alkali. So the titration is actually the experiment through which we can find out the exact amount of alkali which is required to add in acid to neutralize it. So that's why it's an experiment through which we can find out the exact amount of alkali which is required to neutralize an acid is called titration. Another way or another definition, titration is a slow addition of one solution of known concentration, okay, to a known volume of another solution. One solution is alkali, another solution is acid of known concentration until the reaction reaches neutralization, which is often indicated by a color change. It means when the solution of acid and alkali are neutralized, when acid and alkali are mixed together after the reaction, their color. In titration, the equipment required to us, there are 3 major things which are required for this purpose. Number 1, pipette. Number 2, burette. Number 3, flask. So look at the figure. Here we have pipette. Number 2, burette. This is the burette in the third figure, which is C. And number 3, flask. So this is canonical flask, in which is shown in figure A, B, C and D as well. So these are 3 major equipments required for titration: pipette, burette and flask. Now question is this: what will we do? If you remember the previous reaction, the first method, as I told you earlier, through first method we can discuss about titration, and that when hydrochloric acid, HCl and sodium hydroxide, which is NaOH, behaves an alkali, both acid and bases are added together, they gives us salt. So here HCl and NaOH, when they react together, they give NaCl and H2O. Keep in mind, this is a double displacement reaction. We discussed the displacement reaction in previous. Now let's discuss how the reaction will occur, through which we can understand about their concentrations. I mean the concentration of acid and the concentration of alkali. So in the first step, we will use pipette to get the exact amount, or to put the exact amount of acid solution in the canonical flask. It's up to you whether you have to put acid into the flask or put alkali into the flask. You can, here in the figure, by using pipette, we put acid solution, a fixed amount of acid solution, a constant or measured amount of acid solution is placed inside the canonical flask by using pipette. Now this is the first step. Now in the second step, we have to put a few drops of indicator, and which by using pipette into the same acid solution. Okay, and keep in mind, this indicator is actually just used to neutralize, just used to change the color. So it means indicator changes the color of the solution. These are the 2 steps. Now in the third step, place acid in a burette. Here, if you put acid solution in that, I told you, now you can put definitely base into the burette solution. I told you it's up to you whether you use acid or alkali. So place acid or alkali, whether you put in the burette carefully, and put them by a small amount using the holder, using the hole. Through this hole we can release a few drops step by step, which is in 4, and later it would be the time the level of alkali or acid reduces with the passage of time, and there would be a time the color of the solution changes in figure number 5. So in figure 3 we put a small amount of, or the fixed amount of acid or alkali, whatever, and then it will be put into the solution in canonical flask, and in the fifth step there would be a final reading through which we can understand the exact amount of the acid or alkali used in the burette. This is the way through which we can find out the exact amount of acid and alkali during the chemical reaction. So the point is, place acid in the burette. Acid is then carefully added by a small in a, and if we only require the salt, if the only salt is required, the titration is repeated without the indicator, and we will not add indicator during the same process we will do, but on the indicator will not be use, which actually changes color, and we use exactly the same amount of acid and alkali. Now your homework is just to look at the paragraph and the table at page number 130, to which we can understand the color of indicator used for different amounts. So you should memorize this one as well. Okay, thank you very much. I hope you learned a lot from this video. Stay blessed.